All right, everyone, welcome to Module 4. This one's going to be a little bit of an easier lecture, kind of a quicker lecture, because we're going to be covering arthropods and then clinical laboratory procedures. So arthropods are generally going to be our vectors, so that's why there's not as much to cover about them. So again, we're going to be covering Chapter 6, Arthropoda. Arthropoda is an phylum, and it contains more than 80% of all animal life. Arthropods directly cause or transmit more than 80% of all diseases, and they are major economic importance to agriculture with both beneficial and destructive effects. So again, here are your learning objectives for you guys to go through and review. Okay, so the arthropods contain many members and are divided into 5 different classes. 3 classes that we worry about in parasitology are insecta, arachnida, and crustacea. These contain most of the medically important arthropods. So the class Insecta contains many insect types whose bodies are divided into 3 distinct regions: a head, a thorax, and an abdomen. And I have a picture of this on the next slide, I think. The next class is going to be Arthropoda, and these contain, Arachnida. And Arachnida contain our ticks, mites, spiders, and scorpions. And then the next class we're going to cover is Crustacea. Crustacea includes crabs, water fleas, lobster, shrimp, barnacles, and wood lice. And the general body structure again are going to be um, like divided into 3. So the head, uh, the thorax, and the abdomen. So here we just have a general picture of the insecta. So here you see the head, the thorax, which is the central structure, and then the abdomen. So the life cycle. All arachnida and most insects develop from an egg into an adult by a process called metamorphosis. I'm sure most of you guys have heard of metamorphosis in your elementary biology. We could have incomplete or hemimetabolous metamorphosis, and this metamorphosis, again, incomplete metamorphosis, has 3 stages. It's going to have an egg stage, a nymph stage, and an imago stage. So the egg is going to develop into a nymph. And what a nymph is it just resembles a small adult form of the egg, and then the imago is the sexually mature adult insect or arachnid. After each molt, the nymphal form is called an instar. Wings of flying insects increase in size with each instar. Lice, true bugs, and arachnids develop into metamorphosis. In a complete metamorphosis, we have an egg, a larval stage, a pupa stage, and an imago stage. The pupa stage or pupae stage is going to be like an encasing resting stage between the larva and imago stage. So in a complete metamorphosis, we're going to include the cocoon stage that you often will hear about with butterflies. So with most crustaceans, we have incomplete metamorphosis, and the stages are going to be a little bit named a little bit differently. We have eggs are going to hatch and release free swimming Nautilus larvae, and these are going to molt several times to become mature adults. You could find the life cycle of arthropods on page 123 of your book, and I highly suggest that you take a minute to review the differences between a complete and incomplete metamorphosis. So some of our insects are parasites of the human. So we have ectoparasites, which are going to be like our lice, mosquitoes, and ticks that feed on the blood. But then we also have other insects that, play a role as a vector. And these insects are going to be the ones who are transmitting a variety of diseases. So we could have a mechanical transfer. Insects can passively transfer infective organisms from feces or contaminated soil to food or utensils when they walk across or feed on inadequately protected food. This form of infected transmission type is called mechanical transfer. So, whenever you see flies or ticks on your food, or if you're having a picnic outside, that's can be a mechanical transfer of a parasitic infection. A mechanical transfer implies that the insect is not required as part of the parasite's life cycle, and that no further development or growth of the parasite occurs in the insect. But the insect is just mainly meant to mechanically transfer the parasite. We also have a biological transfer. This is where a biological vector is required as part of the parasite's life cycle because growth and development of the parasite must occur inside the insect. So insects can serve as a definitive or an intermediate host. Infective parasites developed in an insect generally are injected directly into a new host, or in a few species are deposited on the host skin. So the biological transfer, we've heard about this a couple of times now in previous chapters. Most recently, we've heard about the vector transmission of utilizing Ioxides scapularis for the Babesia species infections, blood infections. So insects to parasites. So in addition to mechanical or biological disease transmission, most of these insects um, don't just cause they're not just annoying. They cause a delayed or immediate hypersensitivity reaction to blight bites. In some instances, such as like a bee sting, this can induce a fatal anaphylactic shock or anaphylaxis. We could also get injected with toxins or venoms that could be fatal for us as humans. We could also lose food crops, animal weight gain, and productivity from meiosis. So insects as parasites, so bites and stings. Here we get a mechanical trauma. It's caused by a variety of insects, including bees, wasps, ants, spiders, scorpions, centipedes, and millipedes. Some people have severe allergic reactions to bee and spider venoms, and these venoms can cause anaphylactic shock again. Wasp, hornets, and bumblebees are capable of multiple stings without losing their stinging apparatus, whereas honeybees can only sting once because they have a barbed stinger that detaches from the body as the bees fly away. So honeybees are generally docile, which means that they don't really attack unless their hive is threatened. And then we have a more aggressive bee known as an Africanized or killer honeybee that attacks whenever, and these bees pose greater threats to humans. We also have different spiders that can infect humans. We have the black widow spider and the brown recluse violin spider. The black widow's neurotoxin rarely results in death, but it confers lasting immunity against future bites. But it can produce several complications, including abdominal cramps, hypertension, reduced heartbeat, and a feeble pulse, shock, convulsions, and delirium. The brown recluse causes painful spreading, slow healing, necrotic wounds that require medical attention. Very large doses of venom can cause hemolytic anemia, fever, jaundice, hematuria, and death. We also have scorpions that can sting. Scorpion stings are mostly dangerous to children younger than ages of 5. These symptoms can uh, include pain, numbness, muscle spasms, blindness, twitching of the toes and fingers. Sometimes we could get patients can die from respiratory paralysis. Centipedes and millipedes, as gross as they look, they're not really considered dangerous, but some centipedes can inflict painful bites, and some could actually secrete. Millipedes can secrete a fluid that's going to cause a burn or discoloration of the skin. We also have bed bugs. Um, I'm sure you've all heard about bed bugs at some point in your life. These bugs can cause red, itchy bites and clusters on exposed skin. We also have something that's called myiasis. This is where the larvae or maggots of non-blood-sucking flies invade the tissue, and it's caused a myiasis. So the fly is going to deposit their egg or larvae directly on a skin, uh, and these skins are going to form a myiasis, which is essentially going to be where the maggots are going to be able to feed on the host and burrow into the skin. We have different ways of controlling insects. It's kind of difficult. We could have chemical sprays that are used widespread. Uh, We could include natural predators that are going to feed on the eggs laid by these insects. We can destroy their breeding grounds. We can control them biologically using pheromones to trap them and kill them. There's a variety of different ways to control the arthropod population. So we're just going to get into a class of insecta. We have the class Insecta, which includes various family members. Um, the class Insecta includes the order Diprotera, which includes our flies and our mosquitoes. So this is the greatest medical importance order or class because this is where we're gonna see our blood-sucking mosquitoes and our flies, and these flies and mosquitoes can transmit many viral, protozoan, and helminthic diseases, as we've discussed in previous lectures. These organisms can be differentiated based on their presence of an antenna. Um, they could also have mouth parts, and these mouth parts are going to be for piercing skin or sucking fluid. They could have a coloration and hairy distribution on the body, and then their size and shape of the body of each of the body segments are a little bit larger. So we have two pairs of wings, and they go through a complete metamorphosis. Some of the most important flies that we're going to hear about are the Anopheles fly or the sand fly. I mean, Anopheles mosquito. Um, and then we're also going to think about flies like the sand fly or the seat fly. And we're going to hear more. We heard about these in the last lecture. I'm sorry, I skipped ahead to this lecture because I knew it would be a lot faster. But we're going to hear about this in the protozoan lecture, or you should have if you're following the class in order. Um, I'm not going to go through all the classes. You just need to know that we have flies, mosquitoes, bugs, lice, and fleas that can infect humans. And these species, I'm looking through the book here. If you review pages 121 and 122 in your book, these are actually going to break down the different arthropods and their transmission associations. So you're gonna hear about the Anopheles fly, and these can cause malaria. You're gonna hear about the phlebotomist sandfly. These are gonna cause can spread Bartonella, but they're mostly known for spreading leishmaniasis, leishmania. So I think that you guys should take the time to really look through table 6-1 on pages 121 and 122. Here we get into our ticks and mites. We've heard about these a few times already. Again, I want you to refer to uh, pages 121 and 122. Here, I just included a stock picture of the various ticks that are going to cause human diseases. The one that we hear about the most is the Ioxodes scapularis. This tick is going to spread Babesia, It could also spread Rocky Mountain spotted fever. So those of you guys who took microbiology, refer back to Rocky Mountain spotted fever and think about which organism causes that disease. This is a picture of a sandfly. You note that it has some hairy projections here, and you can see that it's taken a blood meal already. And then here's a picture of a louse. And I have an interesting story to go with this. Oftentimes, as medical laboratory scientists or technicians, we don't think that we're going to come across something like a louse or lice, or it's not something that actually should come through the lab because generally, if a doctor is going to find one, they know what it is and they're going to treat it. But we actually had a doctor in Spirit Lake that found a pubic louse, and he brought it into the lab, and we looked at it under the microscope, and it looked exactly like the one that's on your screen here, and we could actually see the blood from the blood meal that it took from the pubic area, and the blood was cycling throughout the louse, and you could actually see it on the slide, so that was pretty cool. So just note that it is possible to see lice in the lab, or a louse. So the next chapter that we're going to cover is chapter 7, and chapter 7 highlights a lot of the clinical laboratory procedures that we do in parasitology. Um, Again, I'm not going to go into too much detail about the procedures because as we have gone through the various lectures, I explained a little bit about the procedures. As medical laboratory technicians, you're not going to see or perform a lot of these procedures yourself. They're either going to be done by a doctor or a reference laboratory, especially if you stay in North Dakota and work in North Dakota. So the first thing we're going to hear about here is a macroscopic examination. So again, the process of a macroscopic examination is just going to be examining the stool for parasites macroscopically. So we're gonna uh, look at a stool sample. We're gonna note the consistency of the specimen. If it's gonna be mushy or liquid stool, suggest a possible trophozoite or intestinal protozoan. Protozoan cysts are found more frequently in formed stools. Helminth eggs and larvae may be found in either liquid or formed stools. We're gonna examine the surface of the top and bottom of the specimen for parasites. We're gonna look for tapeworm, proglottids, or less commonly, adult pinworms. From there, we're going to break up the stool with applicator sticks to check for the presence of adult helminths. So that's going to be our scarce species. Then we're going to examine the stool for blood and mucus or both. Fresh blood is going to be bright red and it indicates an acute lower intestinal tract bleeding. Bloody mucus suggests ulceration and some sort of material that should be examined microscopically for trophozoites. We could also recover tapeworms, and we want to make sure that we recover the scolex. So we're going to treat the feces with a drug. Adult worms, if they're recovered, they should be examined directly. Tapeworms, we're going to examine the gravid proglottis. And again, we're going to want to follow that left to right examination pattern. Oh, I guess sorry, that's more for microscopic. So let's get into microscopic next. So with a microscopic examination, we could have something where it's a direct wet mount, and this is where we're going to use fresh liquid or very soft stool, and we're going to prepare using applicator sticks. We're going to want to thoroughly mix small amount of feces with a drop of saline, and we're going to want to place it on a microscopic slide. Generally, this is how we're going to look for eggs, larvae, and protozoa on our slide, and we're going to want to follow the left to right examination whenever we report direct wet mounts. We're going to want to report uh, um, both the genus and the species if we can identify the organism based on the egg or the trophozoite. We could also comment on cellular elements. So if we see blood cells, which should be reported. and yeast, so we could report semi-quantitatively, so like few, moderate, or many red blood cells, white blood cells, yeast. We could also report things like Charcot Leyden crystals. Again, we're going to want to report as few, moderate, or many. It's important whenever looking on a microscopic examination that we know what's artifact and what's an actual parasite. And this is something that you'll be um, taught on the job site if you are ever working on my parasitology bench. I'm not going to get too in depth with these examinations because generally, if you guys are going to be looking at an examination, you're just going to prepare a direct wet mount and you're going to comment on if you see something or if you don't see anything. It's going to be very rare that you can do a fecal concentration or staining procedures. Okay, so a sedimentation method of concentrating an organism. This method concentrates parasite stages present in large amounts of the feces. So here we're going to want to take 5 to 15 grams of fresh feces into a suitable container using applicator sticks, and we're going to mix it with 10 to 15 milliliters of 10% formalin. This mixture is going to stand for 30 minutes to allow for fixation, and essentially what happens here is it kills or preserves the protozoa larvae and most eggs. Some eggshells, for like a scarce species, are going to be impervious to formalin, so it's not going to kill the eggs or make them preserved. We're going to want to strain the mixture through 2 layers of dampened surgical gauze, and then we're going to want to centrifuge the specimen. We're going to decant the supernate into the disinfectant, and then we're going to resuspend the sediment in saline or formalin. From there, we're going to centrifuge again, and uh, we should get 4 layers here. What we're going to want to do is we're going to want to take that bottom layer because that sediment is going to contain the parasites, and then we're going to use that sediment to do a direct wet prep. Again, I'm not going to get into too much detail. These are procedures that aren't used as often. You guys aren't going to have to do them, but it's just to note that these are some of the procedures that are taken um, whenever prepping stool samples for examination. Most of the time, you guys are just going to have to take a sample and send it to a transport lab, and the transport lab is going to perform all of these staining techniques and so on and so forth. One test that you guys might actually have to assist with is the cellophane tape test for a pinworm, and I covered this test in a previous lecture. Actually, I think this was lecture 1. So we're just gonna skip over this because we went over it in lecture 1. I would recommend that you go through the table 7-2 on page 151. And what this does here is it's going to tell you how to process various specimens. So like for our Giardia species, we could do entero test from the duodenum, and what essentially this is a wet mount. But these wet mounts are going to tell us where we're going to get our samples and what possible parasites are going to come out of these samples. One procedure that should be relatively easy for you guys to remember is a blood smear preparation. And here we're just going to do it as if we were stating, setting up a regular blood slide for a differential. We're going to create a blood smear. We're going to take that smear and we're going to stain it. And then we're going to go through and do a examination on the microscope. For whenever we are ready to report, we're going to report it as malaria, Babesia species, trypanosomes, leishmania, and we're also going to comment on the appearance of the red cells or if the parasite is inside the cell or not. So it's important to note that we have various types of immunodiagnostic methods in parasitology. Um, we have tests that can detect Cridosporium species. We have tests that can detect Giardia lamblia. The most commonly one that you're going to come across is the kits that are going to help look for Trichomonas vaginalis species. And essentially, what these are going to utilize enzyme immunoassay tests or ELISA techniques or DNA probes and DNA amplification. And these techniques essentially are just going to help us determine if we have a current infection because of the immune response that the body created from this parasitic infection. There are some potential for error with these immunological tests. So some uh, some places are going to do immunological tests, but they're also going to collect a stool sample or a wet prep and utilize that alongside of the enzyme immunoassay testing. I would refer you guys to table 7-3, 7-4, and 7-5 to look through the various testing. So like would be Babesiosa, the organism is Babesia micro T, and we could do an immunofluorescent testing. We could have Chagas disease caused by Trypanosoma cruzi, and here we're going to do immunofixation, immunofluorescent testing trichinosis or Trichinella spiralis. We could do an enzyme immunoassay technique. So it's just important to know that there's just different techniques out there for us to utilize. That's it. I know I didn't cover a lot in chapter 7, but as medical laboratory technicians, you're not going to be doing a lot of parasitology methods in the laboratory aside from immunoassay techniques and direct wet prep techniques. So as long as you know what you are looking for and you are able to prepare a wet prep with basic saline, I think you'll be fine for parasitology. As far as the immunological testing, it all depends on what laboratory you're working at and what their procedures are, and they'll train you on that as you get employed there. But for the most part, at your clinical sites, you're just going to be collecting a stool sample and it's going to get sent to a reference laboratory for an ova and parasite exam. One of the only things that you could possibly do at your clinical site is going to be a direct wet prep from a vaginal sample, and there you're going to see Trichomonas species, and that's it. So uh, let's just review the tables in the chapter, do your chapter outline, and just think that you'll get trained whatever procedure your hospital is going to utilize.